Traditionally, cell culture has been undertaken growing cells on a very flat surface, either originally a glass surface and now more recently a plastic surface. But of course, the flat surface in no way resembles the surfaces on which cells grow in the body. So there's an interest now in mimicking how cells grow in the body, which means growing them in 3 dimensions. And so that essentially is the argument for doing more and more biology in 3D. Now, a spheroid is simply a ball of cells that's come together, and that's probably one of the archetypal modes of 3D cell culture. And in fact, if you think about it, it resembles a tumour, because tumours are also balls of cells growing in 3 dimensions. Increasingly, as the field gets more complicated, we start to grow cells in architecturally arranged areas such that they start to resemble organs, or simple organ structures which we refer to as organoids. So the field is moving very, very quickly now as people want to do biology, disease research, drug discovery, not just in 2 dimensions, but increasingly in 3 dimensions. The major advantage is, of course, the resemblance of the cells as they reflect their position within the human body. And because of that, then the action of drugs is very similar to what one sees in the body when looked at them in 3D versus 2D. And in addition to that, you can start to, even in some cases, use cells derived from the patient, where you can now use patient-derived cells grown in an authentic environment in 3 dimensions. So they're proving to be very powerful disease models. But increasingly, it's turned out that the action of drugs also is very reminiscent of what one sees in the patient. And in fact, there is now extensive data to show that early leads for drug discovery found in 3D resemble what one sees in the patient, as well as some of the metabolism of drugs in 3 dimensions is very similar to the patient as well. In other words, the physiological relevance is increasing, particularly for drug discovery. I would say the field is in transition, so certainly disease modelling, particularly in the areas of cancer, using spheroids is now becoming a standard technique, because the spheroid is often a very good surrogate for, say, a solid tumour there. But increasingly in other areas of disease research, let's say Alzheimer's disease or Huntington's disease, then an organoid culture is now being seen as one of the ways to go. Because the brain is a very complicated organ, and if you can get a model of that in an organoid, you can also have a very good disease model as well. Let's sort of take it from the drug discovery process from start to finish. So the disease model becomes a very important area of research. How can I mimic the disease in a 3D cell culture area? Because if I can mimic the disease, I can identify a drug target, or I can find out more about the pathology of the disease. And again, doing that in 3 dimensions is turning out to be a very powerful way in which you can do that. And that's occurring with a range of diseases. Everything, as I mentioned, from cancer through to heart disease, through to liver disease, through to retinal diseases that cause blindness, right the way through to central nervous system diseases as well. And I would say, by the way, that one of the advantages, you can look at these cells in 3D over time. So in fact, some people have called this 4D culture, the 4th dimension being different time points with the same group of cells. The fact that you can use patient cells to grow into 3D culture means you're exactly in the area of disease modelling with authentic patient cells in an authentic environment. And then when you move those cells into drug discovery, you can use them in high throughput screening, and there are systems developed from companies such as Corning and other companies where you can now do high throughput screening of cells in 3D. And there you can look at many, many compounds in a day. 1 million compounds a day would not be unusual when looking at them in 3 dimensions there. And then as you then move into developing the compounds to go into the patient, Then you're very interested in saying, can I predict metabolism? Can I predict toxicity? And how can I get that to be more reflective of what I'm going to see in my patients? So lead optimization is an area that's now also increasingly being looked at in 3D. The liver cells, for example, appear to be very similar to what one sees in the patient when one studies them in 3 dimensions there. So really, from basic research, disease modeling, drug screening, lead optimization, all appear to benefit by doing these techniques in 3D as opposed to 2D. The most rapid adoption has been in the area of cancer, and in fact that's where this field really started, where probably about 10, 15 years ago researchers were developing techniques to take tumour cells, grow them in 3 dimensions, and the ability to grow them in 3 dimensions completely mimicked how the tumour developed in the body. So a lot of techniques were developed in oncology labs. Then that moved into, can you take it into high throughput techniques? And there's certainly now an increasing adoption of this in terms of automated robotic screening, where you do have automated liquid handling applied to these cells in 3D. And as a result of that, you can screen, as I said, many millions of compounds a day there. Spheroids in particular are ideal for this. And in fact, Corning has systems, as well as other companies, where it makes it very easy to do the growing of the cells, the assays of the cells, in the same plate, in the robotic system, such that everything works very well from start to finish there. And then increasingly, as I said, looking at them in terms of measuring metabolic liability of compounds in cells, liver cells for example, that's increasing as well. So basically oncology for sure, neurosciences subsequently, and then drug discovery now as well. So not every cell will naturally grow in 3D, though many cells grow naturally in 2D, surprisingly, since they all grow in 3D in the body, right? So some cells will naturally form a spheroid system, that is, they'll attach to each other, form a ball of cells very, very easily. Other cells will take different growth factors to drive them together, or different extracellular matrices by which they can come together as well. And neuronal cells in particular are ones that are very finicky in terms of the right cocktail of growth factors, the right environment by which they're going to come together. Liver cells almost every time will form a ball of cells there. I think when you get into organoids, the area becomes much more complicated, because there you're trying to recapitulate development and differentiation of cells into an organ. So they don't just have to grow, they have to grow in the right place. So they've got to be architecturally arranged, spatially located in the correct area that mimics the organ. And you can imagine with a brain organoid, you've got a very complicated organ you're trying to mimic there as well. And that requires a lot of work in terms of optimization there. Now there are some very straightforward extracellular matrices available. Matrigel is one of them, which is a product that's been around for 50 years. And Matrigel is a naturally derived extracellular matrix, and many cells will form an organoid in that particular structure. And then there are others now being developed that can do the same thing for other sorts of the cells. The final point is how long do you leave them growing in 3D? Because if they grow in 3D, then it's just that, a 3D structure. And oxygen penetration can change according to which level of the cells you are looking at. And you can start to get cell changes depending upon their location in the organoid. So the timing of when you look at these cells in 3D is very important. And as we talked about with 4D culture, you may be interested in that anyway. You may be interested in how these things are changing over time. So a lot of protocol optimization is required the more complicated the system is you're developing there. We would recommend using microtiter plates that have an ultra-low attachment surface. A ULA plate is how it's referred to. And often starting the experiments in a 9 to 6 well microtiter plate enables you to take cells grow them for a 4 to 8 hour period, they will not attach to the well, but they will attach to each other. And then over a 2-day period, you'll find that they will generally form a ball of cells or a spheroid. The advantage of working in a 96 well plate is that that's immediately compatible with fluid handling robotics. And because there is no separation step, you can then do an assay in the well in which you've grown your spheroid. If you're then concerned about, I'd like to do higher density plates, in fact you can do the same experiment growing cells in higher plate densities, right the way through to some very high density plates where there's 1536 wells on the plate. And that means you're in very small fluid volumes, but you're in very high numbers of assays per plate there as well. So that would be a good place to start, I think. I think that enables you to do the cell growth and the assay within a single plate, and it's certainly amenable to high throughput technologies. With organoid growth, growing organoids in Matrigel with a variety of different growth factors on board generally enables you to get early cellular differentiation into the organoids. And as I said, liver cells will work very well. Myocytes to form cardiovascular organoids will work very well, as will kidney cells as well. So some of the latest advances are very much around how you analyze the cells in 3D. So one of the very powerful advances is in imaging technology, because if you can use a confocal microscope system to look at the cells, you can look at them in 3 dimensions. And that's really powerful in terms of knowing where the cells are, where the proteins are within the cells, and how they're changing as they grow in 3 dimensions. Now, of course, some of these imaging technologies require incredibly complicated software to reconstitute the image. So in parallel with the growth of 3D is the growth of imaging and imaging associated software there as well. Other advantages come within the kind of systems that they're grown in. So for example, some systems are being developed that will allow high volume production of spheroids, because the demand is now increasing. So within one culture flask, you need to grow millions and millions of spheroids there. Other developments are coming in the area of synthetic polymers to grow cells in a surface using a totally synthetic polymer, like a hydrogel, which is 80% water. Often that requires sophisticated polymer chemistry to develop something that completely mimics what you find in vivo as well. So it's an interesting field from a technical point of view, because you've got developments in engineering, developments in polymer chemistry, developments in photo optics, as well as developments in biology, all coming together to form a very new field there. And it's very exciting as a result.